My name is Melissa and I'm a student at the University of Warwick about to go into my final year studying biological sciences. I spent 8 weeks at the RHS garden Wisley studying the dynamics of plant parasitic nematode genera in gardens. Plant parasitic nematodes are known to be important agricultural crop pests, and lots of research focuses on the resultant reductions in crop yields, but there has been very little research done on the impact of nematodes in gardens. There are reports of over 2,000 investigations into plant and soil-borne nematode genera present in the RHS garden Wisley, dating all the way back to the 1920s, but their occurrence has never formally been investigated. I applied for this project because plant health is an area I had never encountered before or been taught, and I wanted to expand my breadth of knowledge as much as I could through looking into an unknown territory. I also, as a result of COVID-19, had a lack of experience and imperative skill working in the lab and using certain equipment, and I wanted to gain a better understanding of how to navigate a laboratory independently and efficiently. The first step of my project was learning how to identify nematode symptoms in the garden. Following this, I extracted plant parasitic nematodes from soil samples, leaf tissue samples, and also roots, and identified them underneath the microscope at both high and low power. I practiced making permanent slides of the nematode species Aphelinhoidus to identify them to a species level. Following this, I confirmed my identification using molecular techniques for foliar nematode Aphelinhoidus and the northern root knot nematode Melanogyne hapla. My results indicated a high diversity of plant parasitic nematodes in the garden, which were present on many host plants in a multitude of areas across Wisley. The results from the molecular work showed that only one of the protocols is suitable for nematode DNA extraction, whilst the PCR confirmed the previous morphological identification. I have learned a huge variety of skills during my project, including how to do a DNA extraction, PCR and gel electrophoresis using species-specific primers, how to make permanent slides, how to identify nematode infection symptoms in the garden, and how to navigate a microscope to identify different nematode species under both high and low power. This project has set me up extremely well for my final year at university and has given me the experience and skills I need for my final year dissertation and also my future masters. I am so grateful towards the BSPP for granting me a bursary to explore a really interesting area of science I would not have otherwise investigated. This experience has been overwhelmingly positive, and I would strongly recommend exploring the area of plant health to any undergraduate students pursuing a career in science.